In this unit, we're going to talk about muscle. Muscle is very closely related to the skeletal system, so be sure that you're familiar with the skeletal system and all of the bone features before you start to look at the individual muscles. We'll describe the major functions of muscle tissue. We'll explain how the muscular system contributes to thermoregulation. We'll describe the structure, location in the body, and function of skeletal, cardiac, and smooth muscle. We'll compare and contrast the general microscopic characteristics of skeletal, cardiac, and smooth muscle. We'll describe the organization of skeletal muscle from skeletal muscle fiber to whole muscle. We'll name the connective tissue layers that surround each skeletal muscle fiber, fascicle, entire muscle, and group of muscles. The main function of muscle tissue is movement, moving the bones, rhythmic beating of the heart, constricting and dilating tubes such as the blood vessels, gastrointestinal tract, and lung bronchi. Muscle tissue also produces heat as a byproduct of metabolism. If our body temperature falls, muscle contractions provide extra heat through shivering. Muscle tissue in the body is divided into three categories: skeletal, cardiac, and smooth muscle. And you can see those here. Here it lists what order they're in. So it goes skeletal here, so skeletal, smooth, and cardiac down here. Skeletal muscle is striated in appearance and voluntary in action. It's found attached to bones by tendons. So we've looked at these before, but to review, remember skeletal muscle here at the top is striated. You can see those bands in the tissue. And it's voluntary. You think about moving skeletal muscle, you can move it. Cardiac muscle is also striated, and the bands aren't super clear in this picture, but I'll show you a better picture that we also used in the tissue unit for cardiac muscle. But it is striated, but it's involuntary. You don't think about beating your heart. Your heart is the cardiac muscle, and it has its own pacemaker that you'll talk about in 221. Smooth muscle is unstriated and involuntary. So here's smooth muscle. It doesn't have the striations. It forms the walls of the tube, such as the blood vessels, intestines, and hollow organs, such as the stomach. Skeletal muscle has striations when viewed under a microscope. It is also multinucleate. Each cell may have several nuclei, and you can see that down here. Remember that when you're looking at a microscope slide, you're taking a very thin slice through a three-dimensional structure. So if you slice below a nucleus, you may not see it. You'll see it if you slice through it, and there may be one above, but the fibers are actually circular, so you're just making a slice through, like a slice through a loaf of bread. So here we see the nuclei. They're very peripheral, and there may be several per fiber. The striations are formed by organized bands of actin and myosin contractile proteins. And in this picture down here, you can really see those bands. It's nice and enlarged, and you can see the band structure, the light and dark bands. And we'll talk about the myosin and actin and how they form those bands. The bands allow the proteins to align themselves to shorten the muscle lengthwise during contraction. Each band is formed from an organized unit called a sarcomere. Skeletal muscle movements are voluntary. So if we're looking at skeletal muscle, it's muscle that's attached to the bones. That's why it's called skeletal. It's voluntary. The contractions are under conscious control by the brain. The muscles attach to the bones using tendons, and they pull on the bones to move them as levers. We'll talk a lot more about levers, and we'll talk about those individual muscles later in this unit. Skeletal muscle's voluntary control allows it to regulate the timing of inputs to and outputs from the digestive and urinary systems at the sphincter muscles. Now, not all sphincter muscles are voluntary, but some of them are. When we toilet train, we learn to control our anal and urethral sphincters. We also can control the upper esophageal sphincter to some extent for swallowing. So there are ones that we are able to control. And striated fibers are that component of skeletal muscle because of those sarcomeres. Skeletal muscles maintain muscle tone subconsciously, allowing the body to maintain posture and body position. That also is something an infant has to learn. They have to learn how to control their core, both anterior and posterior, to stabilize the body so they can sit up, so they can learn to walk. A lot of that is learning to stabilize the core to gain balance. Tone also supports the abdominal wall and pelvic floors to support the internal and protect the internal organs. Contractions of skeletal muscles, both voluntary and subconscious, contribute to the generation of body heat. Now, the subconscious sometimes can throw people off, but you said the skeletal muscles are voluntary. They are, but you've all experienced in or subconscious skeletal muscle contractions. Shivering is one. If you get a tick, like a tick in your, not a tick like a bug, but a tick like a muscle tick in your eyelid or something where it like flexes, that is subconscious. Hiccups to some extent are subconscious too. Skeletal muscles. Like skeletal muscle, cardiac muscles are striated. So in this picture, you can see those striations a lot better. So you can see the banding, the light and dark banding. And then also remember they have that intercalated disc we talked about in the tissue unit as well. Unlike skeletal muscle, cardiac muscle cells are branched. They're connected to one another by those intercalated discs, and they have only a single nucleus per cell. Again, the nucleus, like look at this one right here. So here we have the intercalated disc. This is a good one. But we have branching there. So we have a branch coming off here. And then we have one nucleus. And again, if it doesn't look like there's a nucleus, it's because of the way they sliced it. They didn't slice through the section that had the nucleus in it. So cardiac muscle is really easy to distinguish because of these intercalated discs here. And once you see them, then you'll always be able to see them. Intercalated discs within cardiac muscle allow rapid ion transport directly between cells to coordinate impulses for contraction. This allows for rhythmic and consistent heartbeats. When the heart beats, we don't want the heart muscles all beating at a different time. That's not efficient. We want it to beat as a whole, as far as it doesn't, the whole heart doesn't beat at the same time. The ventricles, the bottom of the heart, beat at the same time and push that blood out into the arteries. Due to those intercalated disc junctions, cardiac muscle acts as a syncytium, which is a group of cells that function as a coordinated unit. Cardiac muscle is self-exciting and rhythmic. It's stimulated by pacemaker cells that respond to a slow ion leak within contraction. The response is similar to the eruption of a geyser like Old Faithful in Yellowstone. If you've ever seen the geyser in Yellowstone, or if you've seen videos of it, it's very regular. And the pressure will build up, and then the geyser will erupt, and then the pressure will build up again, and the geyser will erupt. And I'm not sure the time span of how long that takes. But in our cardiac cells, the same thing happens. The sodium builds up, and then it generates an action potential. And then the sodium builds up, and it generates an action potential. And it does that about every second. So that our heartbeat is around 60. If your resting heartbeat is higher than that, it's happening more quickly. So the pressure, as pressure or ions in the case of the muscle cells builds up to a critical point, then if it's a geyser, you get eruption. If it's a heart muscle, you get depolarization. But that is regular and it's rhythmic, and that keeps our heart beating. So in a healthy heart, the pacemaker response occurs consistently with every heartbeat throughout the entire lifespan. So when we look at cardiac muscle, they're in the heart. They do have those striated fibers with the intercalated discs, but it is involuntary. It is under the control of the pacemaker cells, not under conscious control. Like all excitable cells, cardiac muscle cells are sensitive to the electrical changes brought about by membrane fluctuations in sodium-potassium. But cardiac muscle cells are also particularly sensitive to calcium ion levels. In cardiac muscle cells, cells enter a plateau phase of depolarization brought about by the actions of calcium ions. So let's look at what that means. So if we were looking at a neuron, and this is kind of a preview, we looked at it a little bit when we looked at tissues. We'll look at it a lot more in the next unit when we look at neurons. But when we look at a neuron, it's typically negative. We'll say around -70. It varies depending on the type of neuron. When it's depolarized, it will when it's stimulated, its charge will change, and it will, we say depolarize because it's getting less polar, it's going closer to zero. So it looks something like this. Sodium in, potassium out, and this bump in charge, it's an electrical change, will transmit that signal along the neuron. If we look at cardiac cells, so cardiac muscle can also carry a depolarization. It looks like it starts out the same. It kind of starts and it bumps up and it starts then to go down, but then it hits this plateau before it goes down all the way. This is still sodium in. This is still potassium out. But we have this other plateau where we're stabilizing that charge at that depolarization level around zero. And it's because calcium is coming in. And that calcium coming in generates what's called a plateau. A flat. A plateau is a flat. So that plateau phase allows for a longer and more sustained contraction, and it ensures the heart does not enter tetanus and seize up. We'll talk about tetanus in a later video. But having that calcium effect, that calcium plateau in cardiac muscle, is something that makes it very different from other muscles. Other muscles don't have that. And it gives the heart some special electrical capabilities, such as regulating the rhythm of that heart by kind of drawing out that action potential, that depolarization. Smooth muscle cells do not show striated patterns. The tissue does have actin and myosin. So when we talk about actin and myosin, smooth muscle does have those, but it doesn't it doesn't organize them into sarcomeres. So it uses actin and myosin, it has actin and myosin, but they do not have an organized banding pattern. In contrast, the smooth muscles have the protein spread throughout the tissue to support contraction in any direction, not just lengthwise. So in striated muscle, the fibers are going to look like this. They're going to be in a banded pattern. In smooth muscle, they're more of an every direction pattern so that they can support contraction in any direction. So we don't see the banding pattern, even though the proteins themselves are there. Smooth muscles typically only have a single nucleus per cell. Smooth muscle tissue exhibits involuntary contraction, so it's not under voluntary control. We don't think about moving our intestines. Visceral smooth muscle is the most common type. Contains cells attached by one another by gap junctions and formed into sheets. The sheet arrangement allows for coordinated contractions and in some body locations, autorhythmicity. So think of the areas where we would need smooth muscle. So this could be a blood vessel, that could be in a tube in the intestine, and we need the blood vessel to constrict sometimes. So let's say it's a blood vessel. Sometimes it will vasoconstrict and make the lumen smaller. So if we're constricting. And sometimes we need to vasodilate and make it bigger. And muscle, smooth muscle, allows us to do that. It allows us to constrict and to dilate the tube. It also allows us to shrink it like an inchworm and can compress it. If you've ever seen an inchworm move, compress, extend, that allows us to move contents through the digestive system. We do that. So coordinated contractions necessary for efficient peristalsis contraction of the contents through the stomach and intestinal tract. Also another place is the ureters. The ureters will push the urine using smooth muscle from the kidneys towards the bladder. In skeletal and cardiac muscle, contraction occurs when calcium is released into the cytoplasm from storage in the sarcoplasmic reticulum organelle. So in the muscle, so this is the muscle fiber here. This is the muscle membrane. It's called a sarcolemma. The muscle fiber will get the signal for excitation, and that signal, that action potential, electrical signal, will travel towards an organelle called the sarcoplasmic reticulum. It's like the endoplasmic reticulum we saw way back in the cells unit, except it's specific to muscle, so it's called sarcoplasm. So that's going to stimulate the release of calcium. Remember, I said calcium is a big player in cardiac muscle. Calcium will stimulate skeletal muscle, but it doesn't cause that big plateau, but it will have an impact on contraction. So upon release, calcium binds to a protein called troponin. So here you can see troponin, and you can see the little calcium ions, so they're little gray circles binding to the troponin. That will move in response and open up binding sites between myosin and actin for muscle contraction. So as long as troponin is bound to the actin, then this myosin can't bind. But once we have calcium, that's going to move it out of the way, and then the myosin can bind to it. So we need calcium to open up that spot for myosin to bind. So we can't contract muscles without calcium. In contrast, smooth muscle does not use troponin. Instead of troponin, it uses a protein called calmodulin. So instead of the troponin, we'd have calmodulin. But other than that, it would be pretty much the same process in that the actin will bind to the myosin and cause contractions. We don't have the sarcomeres because remember, in smooth muscle, we have fibers going in multiple directions. Also, smooth muscle brings in most of its calcium from the extracellular fluid. It has some sarcoplasmic reticulum, but it doesn't store a lot in there. Instead, if this is the cell here, smooth muscle is going to bring in a lot of that calcium through transporters when it needs it. So rather than storing a lot in the sarcoplasmic reticulum, like skeletal and cardiac muscle cells do. Skeletal and cardiac muscle cells store a lot of that calcium because the contractions in cardiac muscle have to be so consistent, and in skeletal muscle have to be so powerful. So it helps them to store that calcium there. Whereas smooth muscle needs the calcium, but it can export it or import it from the external environment when it needs it. Skeletal muscles are highly organized to maximize efficient muscle contractions. Muscles are covered with a dense connective tissue protective layer called the fascia. The fascia encloses groups of muscles outside the epimysium of the muscle. So here's an epimysium of one muscle. When we're looking at skeletal muscle, the epimysium will be surrounded by the fascia. So the epimysium is an outer layer of one muscle. The fascia will actually surround multiple muscles in a group. And then all of those muscles will continue at the end to form a tendon. So the skeletal muscles will come together at the end to form a tendon, and the tendon then will attach the muscle to bone. So the outermost tissue is that fascia that will combine with the epimysium of each individual muscle to form the tendon. So deep to the fascia is a layer of connective tissue called the epimysium then. Epimysiums will surround one whole muscle, so a whole muscle. Within the epimysium is another layer of tissue called the perimysium. So here we have the perimysium. The perimysium is surrounding one fascicle, which is a bundle of muscle fibers. So within a muscle, the whole muscle is surrounded by epimysium. A group of muscles would have been surrounded by fascia. We saw on the previous slide. One muscle surrounded by epimysium, one muscle fascicle within that muscle surrounded by perimysium. In Greek, the word epi means upon and the word peri means around, and traditionally an epi layer is your outermost of an individual structure. Epi will typically be superficial to peri. So peri, it also means around, but a peri will be around a smaller structure than an epi. Epi's bigger, so it will be around a bigger structure. Within the perimysium are fascicles. So here we have the perimysium, and within the perimysium are individual fascicles. So here we have the perimysium. It's laying outside of a fascicle, and here we have a fascicle. So the perimysium surrounds a fascicle. These are small bundles of skeletal muscle fibers, and each muscle fiber, so here you have it, you see it circling one muscle fiber inside of that fascicle. Each fascicle inside each fascicle is more connective tissue, which is true, the endomysium. So here you see the endomysium within. So it's pointing inside the fascicle. The Greek endo means within in anatomy, and endo layer is typically the deepest layer of connective tissue. And it is, because once we get inside of the endomysium, then we're inside of a muscle fiber, which is a cell. So epimysium outermost, then peri, then endo, then we're inside a cell. And then the sarcolemma is the plasma membrane of a muscle cell. There. So the endomysium surrounds the individual muscle cells. Inside muscle cells are the myofibrils. So those are not cells. This is the cell. The myofibril is a bundle of those actin and myosins. So those are thick cords of myosin, or cords of thick myosin and thin actin protein filaments arranged in the sarcomere bands. So that is bulk protein. So the protein that you think of as protein in a muscle, that's those myofibrils. That's what's making it up. That's where those proteins are. That's where actin and myosin are. You can see that the mitochondria pack around the myofibrils to meet the energy needs of the contracting muscles. Each of these is a mitochondria. Lots of mitochondria. Mitochondria can divide on their own, independent of the cell. And so if you use your muscles a lot and you build your muscles and you use them especially for aerobic respiration, you will make more mitochondria, which will make working out easier. So the more you work out, the more mitochondria you make, which makes working out easier. So sarcomeres show up as alternating dark myosin and light actin regions under a microscope. In the next video, we'll look more at sarcomeres and how they function.